Hi, my name is Sébastien Lacube. I am an associate at Phymea Systems. I'm sorry I could not be available today, but I hope the video can keep you interested. We are a young company aiming at building phenotyping systems for plant science. We mostly work with open source tools to provide low-cost hardware. We also develop software and image analysis tools. We have been working on various projects, and the one I'm here to talk about is the project we had in collaboration with INRAE in Montpellier, for which we developed a phenotyping system for high-throughput maize ear phenotyping called the EARBOX. So what is the EARBOX? As you know, to perform statistical and genetic analysis in general, on maize ear phenotypes also, you need a great deal of data. And phenotyping manually 50,000 maize ear is quite resource consuming, both in terms of money and time. So the EARBOX resolved the problem by increasing the throughput and the precision of measurements. It's a dual system composed of an image acquisition station that you can see there, and a software for analysis that allows a higher throughput and more precise measurements than manual estimations. The whole logic of the acquisition system is to acquire normalized images of ears, their whole circumference. For this purpose, they are placed on rollers, and images are taken on 6 sites. If you're familiar with it, the system is composed of 2 Raspberry Pis imaging 6 ears at a time in both the RGB and the infrared spectrum. And this can be customized for 3, 6, or 9 ears at a time for various throughputs. And in these configurations, the system takes 30 seconds for 6 ears. So if you account for manipulation time, putting them together, scanning their code, and taking them out, you end up with around 10 seconds per ear, so around 6 times faster than manual measurements. So something that would take 1 week will only take a day. And for the software itself, as it demands high computing capacity, the analysis is done afterwards, separately. One must take the hard disk from the EARBOX machine and plug it into a computer. From there, we have a software for the analysis to output traits of interest. We have 2 models for clients. Analysis can be performed either on Phymea's infrastructure, after sending us the images, or on an analysis station set up in the lab from which you can directly extract the results. Obviously, licensing can be discussed and fixed depending on labs and infrastructure needs. So which variable do we measure with our software? Well, actually a lot. We've built a machine learning and deep learning algorithm trained on a large phenotypic diversity that allow measurements of all typical variables of the ear, length and maximum diameter, and the grains, such as the number of ranks, rows, number of kernels per rank or rows, and number of kernels per ear. We also provide characterizations for abortion, fertile length, and estimations of basal and apical abortion lengths. For people interested in ear and grain colors and types, we can provide measurements of grain and cup colors based on our in-house classification algorithms. As those are specific measurements that can be dependent on client needs, can also be customized with specific classifications. And finally, with image analysis, we can actually go further, much further than this, and output data of grain individual dimensions and their positions along the ear to study grain development. And we've also developed a new variable that takes cob and grain pixels into account to estimate the grain set ratio that can be used to precisely analyze abortion along the ear. And if you're interested, we have a paper on the way that just came to be revised. So I'll invite you to take the link on the QR code on the poster if you want to know more about the methodology we developed. So how precise are we? We tested phenotypic variables against manual measurements for both development datasets on the left that you can see in our paper, and on the right for new data, 1 point per variety, which was acquired last year with GVS. It precisely shows that you can expect great precision for the outputs, which are comparable to what you can get manually, but with higher throughputs. And one advantage also is that you can avoid experimental effects, that you can compare experiments together, as the machine will always apply the same methodology. And we're actually especially happy with the kernel per ear calculation we have here for both the development and the validation datasets. So what's next for us? Well, we have been busy at the end of the year building partnership to improve our system and to provide new traits and new crops. We have an engineer working together with us and David Rousseau from Laris Lab and University of Angers, in collaboration also with CRAW in Belgium, to develop methodology for acquisition and image analysis for wheat, barley, spelt, and other cereals. And we've built a partnership with Serum in Canada to enhance our maize algorithm and provide recognition and quantification of ear pathogens, such as fusariosis detection, and western bean cutworm estimation of damage. If you want to know more, there's a YouTube video of the EARBOX system we just made, and you can also check the poster and come back to us if you have any question. If you want to test the system, we usually provide a possibility to test it at your place by renting it for a small expense. Thanks a lot for listening and for the invitation. I'm available for any question, and I hope you have a pleasant end of day.